Hello, so today we're going to learn about DNA and its structure, okay? The objective of the video is going to be for you to be able to identify the components of DNA. So at the end of the video, you should be able to do that. Also, you should be able to describe how the genetic information is carried in DNA, or more importantly, where is the information carried. And also, you should feel comfortable recognizing that the DNA is universal to all living organisms, okay? And again, you should be able to identify all the components. If you're given a model or a drawing or a structure of DNA, you should be able to identify those components, okay? Alright, so let's get started. Okay, so first and foremost, DNA is a blueprint of life. Just like the blueprints of a house that a construction worker looks at and uses to build a house to the specific dimensions and measurements that the owner wants it to be. DNA is the same thing. It is the blueprint of life because it has all the instructions of how that specific organism is going to look like. If we're talking about yourself, or about you, or any other human being, then DNA has the structures of all the traits that you're going to be showing, like the color of your hair and how tall you're going to be, and the color of your skin, the color of your eyes. All the traits that you have. Traits meaning all the physical characteristics that you have. Okay, so again, DNA is the blueprint of life, and I guess that's why I put this picture here in blue, because it's the blueprint of life. Okay, alright, so moving on. Again, DNA stands for deoxyribonucleic acid, and the reason it's deoxyribonucleic acid is because one, it's made out of deoxyribose, which is the sugar, and the type of sugar in DNA, and we'll talk more about that specific sugar. And also because it's a nucleic acid. We've talked about earlier about biomolecules, and we said nucleic acids. The two nucleic acids that we talked about were DNA and also RNA. But in this video, we'll focus on DNA. Okay, so again, deoxy because of the type of sugar that's in DNA, and nucleic acid because DNA is a nucleic acid. So again, DNA stands for deoxyribonucleic acid. Okay, and the structure, okay, was discovered or established by two scientists named James Watson and Francis Crick, okay, in 1953. Okay, and this guys determined the structure, and they also used a lot of information or a lot of other research by other scientists like Rosalind Franklin, who was doing X-ray diffraction. Okay, she came up with the image, or not came up with it, but discovered the image of DNA using X-rays back in '52. So they used that structure, that image, to come up with the structure of DNA. So they're very famous for that. And you can see it on the picture here. That's the actual structure that they came up with, and because of the bonds and the way the nitrogen bases, which we'll talk about later on, are in the DNA molecule, okay, it actually creates a specific shape of the molecule, okay, and that's all chemistry. So, but again, these two guys are very famous for that, for establishing that structure, and that's the structure that we still use today, okay. So the structure of DNA, you can see it on the side here, okay, and I know you guys have seen it before, but the structure is a double helix, okay. It's a double helix, and it's got two strands of nucleotides, and we'll talk more about what nucleotides are, but you can see there's one strand here, okay, so that's one strand of nucleotides, and then sorry about that, and then so one is right here. I'll do it again, and then the other one is going to be right here. And so that's the reason it's called a double helix. It's a double-stranded molecule, two strands of nucleotides. We'll describe what nucleotides are in a minute. And also it's anti-parallel, meaning that one of the strands, if you see here, you can also see the arrow. This blue strand is going up, right? It's in that direction, and the other strand is actually going down, okay? So they are parallel, but they're anti-parallel because of the direction that they're moving, or the direction that their molecules are arranged, okay? So that's the reason this is anti-parallel. Now, Also, DNA is always located inside of our nucleus and it never leaves the nucleus. We'll learn more about how that genetic information actually leaves the nucleus later on, but for now we need to understand or know that DNA is inside of the nucleus, okay, which is the control uh, house, right, of our cells, controls everything in our cells. And it's also universal. DNA is universal. It's in all living things, and it stores all the genetic information of the organism, whatever organism you're looking at. Okay, and that's the reason it's called universal. Because if you look at the DNA of a cow and the DNA of a human, they're gonna be the structure is gonna be identical. Their structure is gonna look just like this structure right here. There's some components that are going to be in different order, but the structure of a cow's DNA, the structure of a strawberry's DNA, and the structure of a human DNA is going to be identical. Okay, so DNA is a universal. The structure of DNA is universal, and DNA is in all living things. Okay, all right. Now DNA. DNA is made out of these molecules, and these molecules are called nucleotides. Okay, and we talked about this molecule before, this not molecule, but this unit before, and we said this were the monomers. Okay, monomers of nucleic acids. Okay, so we know that DNA is a nucleic acid, so nucleotides are the monomers that make DNA. Okay, and they're always going to have three components, and I'll show you another picture of it. Okay, they're always going to have one, the phosphate group. Okay, another part that they're going to have or component is always going to be the deoxyribose. That's where the name for DNA comes from, deoxy, right? It's a sugar, and also a nitrogen base. And you guys know that A, T, C, and G stand for adenine. A stands for adenine, T stands for thymine, G for guanine, and C for cytosine. Okay, so those are the three components of DNA. Okay, the three components of DNA are phosphate, deoxyribose, and nitrogen base. One of the nitrogen bases, it could be A, T, G, or C. Okay, now you put a bunch of these nucleotides together, and you're going to end up with DNA, okay, or a polymer. Remember, the nucleotides are the monomers of nucleic acids, and in this case, it's going to be DNA. Okay, all right, and the nitrogen bases are what make up the genetic code of the organisms. This is the nitrogen bases. Okay, is what carries the genetic information. It's where the information of your or all the genetic information is stored in your cells. Okay. So that's why nucleotides are so important, and the order of those nucleotides are so important. Okay, staying with nitrogen bases, pyrimidines are bases that only have one ring, and you can see the picture over here. Okay, one ring. So one ring. They have a lot of nitrogen. If you look at the structure, that's one of the reasons they're called nitrogen bases. There's a nitrogen. Okay, there's a nitrogen. There's a nitrogen. There's another nitrogen. So they have a lot of nitrogen, and that's why they're called nitrogen bases. But pyrimidines have one ring, and if you compare them to purines, which are A and G, okay, purines have two rings. Okay, so that's the difference. C and T have one ring. A and G have two rings, and then the special case here of uracil. Uracil is a nitrogen base that's only going to be found in RNA, and we'll talk more about uracil later on. Okay. But these are the types of bases that DNA has. And we talked about the base pairing ruling, which states that A always bonds with a T, or adenine always bonds with a T, and G always bonds with a C. Okay, that's the base pair rule. And you did your model, or you should have made a model in class, and you can see you color coded your model, and this model right here is also color coded. This green color right here is always with the purple, and we can see that. Okay, and it's the pattern there. Okay, A with T, G with C. Okay, and that's the base pair rule. Always the base pair rule states that all these bases are always going to be bonded together, but more than that, it states that G and C are going to be bonded together with 3 bonds. Okay, and A and T are always going to be bonded with 2 bonds, and the bonds that are used for the molecule are hydrogen bonds, and the reason they're hydrogen bonds is because hydrogen bonds are very weak. Okay, they're very weak, so they're very easy to separate, and that's going to be important when we start talking about DNA replication. Okay, DNA replication requires these bonds to break very easily, and that's the reason why hydrogen bonds are an important component of DNA. So that's how base pairing rule. Okay, so A with T, G with C. Now, the levels of organization, or the way DNA is actually crammed into the nucleus, okay, is by folding on itself. Okay, the first thing that happens is this DNA molecule, or we know this is the DNA, is a double helix, right? Wraps around these proteins right here called histones. Okay, so the DNA wraps around the proteins, which are histones. Once it does that, okay, that structure where the DNA is wrapped around the histone is called a nucleosome. Okay, now these histones are going to keep folding and folding and folding, and eventually they're going to fold on themselves, and they're going to make something called a chromatin or fiber, chromatin fiber. Okay, but they're going to keep folding and folding until they're really, really tight. And once they can't fold no more, then they're going to fold the structure that you've seen before, which is a chromosome. So again, chromosomes are DNA. Okay, it's just that it's DNA that's wrapped around itself really, really, really tight. Okay, and this is the way it happens. DNA wraps around histones. The histones with the DNA on them are called nucleosomes, and then those histones keep wrapping on themselves, okay, and they form chromatin. Chromatin keeps folding, and then that becomes the chromosome, okay. And we'll talk more about chromosomes when we get to mitosis. Now the DNA structure, okay, this is a DNA structure. Two models that you can see, or you should be able to identify, okay. The double helix, you can see the double helix. This is actually not twisted, so it's not like this structure. This structure is twisted, so you see the double helix on that. Okay, the one on the right, well, you can still see that we can separate it. Okay, and we can actually see that this right here, okay, is a nucleotide. This right here is another nucleotide. This right here is another nucleotide, and we said that nucleotides are the monomers of DNA. So again, repeating nucleotides or double-stranded molecule. Here's another nucleotide. Okay, here's another nucleotide. Okay, here's another nucleotide, and we can keep going, right? Here's another nucleotide, and then on the other side we actually have another one. So you can see how the nucleotides, okay, there's two strands of the nucleotide, so it's double-stranded, it's a double helix, and it's anti-parallel. Remember, this one is going in that direction, okay? This one is going in that direction. They're parallel to each other, but they're going in opposite directions, okay? And not only should you be able to identify those nucleotides, but also their components. You should be able to identify that this part right here is a nitrogen base, this is a nitrogen base, this is a nitrogen base, that is a nitrogen base, okay? Also, these other ones right here, these are nitrogen bases. The center part, or the middle of the structure, are the nitrogen bases. A always bonds with T, G always bonds with C, okay? And that's base pairing ruling, okay? And then also on the backbone, which is very important, you're always going to see a phosphate group with a sugar, okay, attached to a sugar, okay? And we can see that here. There's your phosphates, okay? There's always a phosphate, but those phosphates are always bound to that sugar, okay? Phosphate bonds to a sugar. So there's another sugar, there's another sugar. So the phosphate is always bonded to the sugar, and they make the backbone of the DNA, right? And we see it on the other side as well. There is the sugars, okay, and all the sugars are bonded to these phosphate groups, okay, and that's very important because that's what keeps the structure of DNA the way it is, okay. So that's just the backbone. That's what keeps everything together. But what carries the genetic information, again, is the nitrogen bases, okay? And remember that A always bonds with a T and C always bonds with a G, okay? So again, That's where the genetic information is carried, and that's why it's so important that you are able to identify these components. Okay, A and T are always bonded with 2 hydrogen bonds. C and G are always bonded with 3 hydrogen bonds. Okay, now the hydrogen bonds are very weak, and that's a very important thing. Okay, for us as humans or for our cells, because when DNA replication happens, we actually have to separate the DNA molecule, and those hydrogen bonds allow us to do that. Okay. All right. Now, what components make up the genetic code of an organism? We already know that, and you should be able to identify it on this picture, which is the one we just saw. Right. We have our deoxyribose, which is our sugar. We have the nitrogen bases, which are right there. These are also the nitrogen bases. Okay, two different A, T, C, and G, and also our phosphate group. Okay, our phosphate and sugar makes the backbone, and our nitrogen bases carry the genetic information for the organism, or at least for us. Okay, now the combination, like I said earlier, of the bases of A, T, G, and C determine what traits you're gonna have, right? Like if you look at this letters that I have here, right? They're the same letters, CAT, in both parts, right? But this one says cat, cat. This one says tack, tack, because they're in different order, and it's the same thing in our genes or our DNA. So the way that those bases are organized determine the traits that that organism is going to have. Okay, it's those nitrogen bases that carry the genetic information and that determine what that organism is going to have, or the traits that it's going to have. So again, by now you should be able to identify the components of DNA if given a model or given a structure or a drawing. Okay, you should be able to identify the nucleotides, a nucleotide, also the parts of the nucleotide, which are the phosphate, the sugar, and the nitrogen bases, A, T, C, and G. You should be able to identify the hydrogen bonds and why they're important. Also know why they're important. Also, you should be able to, or you should know by now that the genetic information is carried in the nitrogen bases of the DNA molecule, and that the backbone of phosphate and sugar are just there to keep the structure of the molecule, and that DNA is universal, is in all or living organisms, but okay, it's in all living organisms, but the reason we look different, even though our DNA is it's identical in structure, right? The reason we look different is because our nitrogen bases are organized in a different sequence, or there's a different sequence of our nitrogen bases. Okay, all right.